Good morning, Principal Sir, and good morning, my colleagues. I welcome you all for my topic on cyst nematodes. So before entering into the cyst nematodes, I wish to briefly explain about what is nematodes and how it is classified and what are all where it lives and a small history about the nematology development. So first, the nematology, it is the study of nematodes or roundworms. So the word nematode is derived from a Greek language. It stands for nema and oides. Nema means thread and ois means resembling or look alike. It means the nematode, it stands for a thread-like organism which infest on plants and animals. So a small definition of nematodes, it is a small microscopic in nature and its size varies up to 0.5 to 12 mm. The size I mentioned here is only for the plant parasitic nematodes. Okay, so then it is a diploplastic in nature, bilaterally symmetrical. It has sexual dimorphism and it have the biotic systems like digestive system, excretory system, nervous system and reproductive system, but it lacks in respiration and circulatory system because it is a pseudocoelomate animal. So it lives in soil, water and also in plants, humans also and animals also, and in water it lives in both fresh water and marine water. It lives in both the extremes like freezing point and also for hot springs. Sometimes also it lives for freezing point. One nematode I quoted here is Steinernema anemoli. This is an entomopathogenic nematode. It lives under even -4 degrees Celsius at the freezing point. Aphthalmcoides species, one of the Aphthalmcoides species, it lives even in the hot spring season in New Zealand. It is recorded as at 61.3 degrees Celsius. This species lives. And the nematode, it requires only a thin film of water for its survival. So the development of nematology, how it begins means first Borges, he identified the first free living nematode. It called as vinegar eelworm, that is Darbatris asceti, and then Tyson, he identified the human parasitic nematode Ascaris lumbricoides in the year of 1683. After that, the studies on the animal parasitic nematodes was developed. So, the helminthology, it is the branch of nematology and it is used to study the animal parasitic nematodes which the nematodes infest on both human and animals. So Karl Asmuth Rudolphi, he is the person, Karl Asmuth Rudolphi. He is called as father of helminthology, and after 1752, after 19th century years, the development of nematology, especially plant parasitic nematodes, was developed after nematodes identified by N.A. Koch. So he is called the father of nematology. So nematology is the study of plant parasitic nematodes. So the nematodes are generally classified into two categories. One is free living nematode and another one is parasitic nematodes. In the parasitic forms, it comes under both animals and parasitics. And the nematodes, not only for these two categories, the nematodes also used as a biological indicators and pollution indicators, biological control agents, and some nematodes also used as a control of nematodes. So I quoted some references of nematodes, but for the nematode used as biological indicator is Xenorhabdus elegans. Panagralis redivivus, it is used to identify the zinc and lead polluted in soils. And entomopathogenic nematodes, Steinernema and Heterorhabdus, these are two entomopathogenic nematodes which are used as the biological control of insects. And some predatory nematodes also available for the management of nematodes as well as insects. So the free-living nematodes, there is not much studies available for free-living nematode because it does not cause any diseases or impacts side effects on humans or animals or plants. But it lives in both freshwater and marine water. It is very minute in size, and I give some examples also here. Then the parasitic forms of humans and animals, one is Ascaris lumbricoides, another one is Wuchereria bangrafti. These are the mostly widespread human parasitic nematodes because the Ascaris lumbricoides, it causes anemia in humans, and Wuchereria bangrafti, it causes elephantiasis diseases, and the Wuchereria bangrafti is transmitted by mosquitoes. The Ascaris lumbricoides, it spreads due to the poor hygiene. So in world level, up to 25% of people are infected because of Ascaris lumbricoides because of the poor hygienic condition. Then it comes to the plants parasitic nematodes. So it is most economic important in horticulture, agriculture and forestry crops, and these nematodes are identified after the 1975 like that years after invention of animal and some human parasitic nematodes. So the first plant parasitic nematode is Anguina tritritsi. It is identified by Neatham in the year of 1743, and it will infest on wheat crop, and it causes the disease of seed gall. And mostly all the plant parasitic nematodes are microscopic, and it lives in a hidden forms. So soil or plant leaf part or below ground part, roots also it will infest on both the areas, and it does not create any clear-cut symptoms. When compared to other symptoms, diseases or insects or nutrient deficiency, we cannot clearly identify the symptoms of nematodes. So because of these reasons, the separate department of nematology will be developed. So in India, TNAU is the first institute which develops a separate department for nematology in plant protection. So the economic importance of nematodes. In worldwide, the nematodes, it will cause up to the yield loss of 12.3%. In major crops, major consumer food sources, so up to 10.7% of yield loss will be caused by the nematodes. Commercial important crops, major 20 commercial important crops, the crop loss due to the plant parasitic nematode is 14%. In developing countries, the nematode will cause the yield loss up to 14.6%. In developed countries, it causes 8.8% loss. So these economic importance is judged by the four factors depends on the parasitic or potential of the pathogenic ability of the nematode and where it is geographically distributed and the value of the crop. So based on these three factors, the economic importance of plant parasitic nematode is judged. So in these ideas, I hope that you get a small idea of nematodes. So in this category, the cyst nematode, it becomes the most devastating pest of crops. So mostly the cyst nematodes, after root knot nematode, cyst nematode is the primary causal organism of yield losses in many agriculture and horticultural crops. So the first cyst nematode, it was identified by the scientist Jancy in the year of 1859. He identified the nematode in sugar beet crop and it caused the disease of beet tiredness. So he classified the nematode is Heterodira shakti. So after 1940, Franklin, he differentiated many cyst nematodes based on the morphological differences. Now there is 8 genera are available in cyst nematode. One is Globodira, Heterodira, Punctodira, Catodira, Dolichodera, Neodolichodera, Butylodera, Aphanistra, Cephalodyra. These are the 8 genera of cyst nematodes available in worldwide. In that, Heterodira and Globodira are the most economic important nematodes present in India, and these nematodes are host-specific and sedentary endoparasitic nematodes. So sedentary endoparasitic nematode means the nematode, when it turns into a adult stage, it becomes immovable and it present in a single cell and suck the plants up from all the sides of the plant. So there are for analysis or study about the nematodes, first we have to extract the nematodes from the soil. So mostly cyst nematodes are present in the soil. So cyst is a dead and females. So these nematodes are present only in the soil, not in the plants. So we extract the cyst nematodes from the soil sample by 3 methods. One is conical flask method, Fenwick method, and robotic system. So the conical flask method and Fenwick method, these are the 2 conventional methods identified in the year of 1940 by Dr. Fenwick and Erling Meyer. They are the scientists identified these 2 methods. But now, 2021, the State University of Illinois from USA, they identified with the robotic system for extraction of cyst nematodes and eggs from the cyst nematodes. So first, conical flask method, we have to take a small quantity of soil in a conical flask and you have to add up some water up to the neck level. Then you have to shake the soil sample with water and leave it for 10 minutes. After that, the cyst will start to float on the ridges of the water. Then we have to extract into a wire mesh and allow to dry. After the sample will be dry, then only we can observe the nematodes. Then Fenwick method. So in earlier days, the Fenwick it is made up of a brass material, but now it is available in many cultures like glass, plastics also it is available. So it has some parts. This first one is, this part is funnel. Then inside that 20 mesh sieve will be available. Then this is the base. This is collar region. So here we will collect the cyst nematodes in 60 mesh sieve. So what is the procedure for this method is, we have to take the soil sample in the funnel which containing the 20 mesh sieve and we have to apply the water continually for extraction of nematodes. So then next one is robotic system. So in this method there is 4 sieves are used. One is 20 mesh, 60 mesh, 200 mesh and 500 mesh. These are the 4 types of sieves are placed here to collect the nematode, and the procedure is we have to input the soil sample or cyst nematode. So in this one single method we can extract both cyst nematode as well as the eggs from the cyst nematode. So here we can use many steps. For extraction of cyst nematodes, there are 6 steps are involved. Extraction of eggs from the cyst nematode, there is 10 steps are involved. And by using this method, we can extract the nematodes. So the taxonomy of the cyst nematode is phylum Nematoda, class Cetanentia, order Thalassophila, suborder Thalassophila, superfamily Thylakoidea, family Heteroderidae, subfamily Heteroderidae, and the genus is Heterodira, Globodira, because these 2 only mostly wide present in India. Then the small morphological characters for easy identification. The heterodira nematode, the female, it looks in a lemon shape with a short neck and it appears in a brown or black in color. Then the globodira nematode, it present in a spherical shape and it have a globus shape. So heterodira, this word is derived from a Greek language. Hetero means different, hedera means different, and dera means body wall. Rather, the nematode it becomes a thick body wall when it dies. So the globodira it derived from a Latin and Greek word. Globus means round and dera means body wall. So the globodira it has a small difference between the globodira and heterodira is the body shape. One is spherical in shape and the vulval bone is not present in the globodira. So little biology. So before explaining this, I wish to play a video for easy understanding of biology. Video. So how the nematode will emerge from the eggs and how it will infest, how it cause the disease. So this is a brief biology about the nematode. So this is the cotyledon stage of the plant and the nematode it will start to emerge at this stage itself because it already present in the soil. When it get the stimulants from the plant, the nematodes will emerge from the eggs. So this is the cyst and this is individual eggs. From the egg, the second stage juveniles of the nematode will emerge from the egg. So the small dots, it refers the chemicals which released by the plant. So the second stage juvenile will emerge out from the cyst by breaking the vulval region of the nematode and it enters into the plant cell. So this is the stylet. So by using of this stylet, it will enters into the plant. So first it will enters in the cortical region, then finally it moves up to the xylem phloem vessels also. So it releases the cellulase enzymes to dissolve the plant cell walls and enters into the plants. And once the nematode it reaches the xylem phloem vessels, it will find its feeding site. So after that it will insert its stylet into the plant cell and suck the saps from the nearby cells and dissolve the cell walls of the plant by releasing the enzymes, salivary enzymes of the nematode. So these yellow and orange particles are mentioning the salivary enzymes of the nematode. And then the nematode it will become immovable, that is mentioned as a sedentary endoparasitic nematodes. And it will freeze the cell contents by stimulating the chemicals, and after that the cyst nematodes it will connect 10 to 12 neighboring cells of the plant cell and produce a feeding site it called as syncytium. So from this syncytium it derives all the nutrients for its further growth and development. Patchy appearance, reduced the leaf lamina size and yellowing of leaves, the flowers and parts of, because the heterodira and globodira, the globodira it specifically it will infest on the potato, not other the crops, but it will infest only the potato crop, and the heterodira it will infest on the pulses of the crops. So it will reduce the flower size and part size after it will infection, and it produce a poorly developed root system. It causes a purely white symptom. It means the nematodes will attach with the roots and it shows a pure like முத்து கோத்து வச்ச மாதிரி அது வந்து இருக்கும். So it is called as pale root symptom. So the economically important cyst nematode species is Heterodira avinae. I already mentioned the both Heterodira and Globodira, they are host specific plants. So individual species will infest only the specific crops alone. So the cereal cyst nematode Heterodira avinae it will infest on the cereals of the agriculture crops like rice, other than these. Then Heterodira glycine it will infest on soybean. Heterodira gajani it will infest on pigeon pea. Heterodira zeae it will infest on maize. Heterodira trifoli it will infest on clover. Heterodira cruciferae it will infest on the brassica family crops. And Globodira, in that Globodira there is two species are available. One is Globodira rostrocyansis, another one is Globodira pallida. These two nematodes infest only on the potato, and this nematode becomes a quarantine nematode. So for Heterodira avinae, this nematode was identified by Dr. Kuhn in the year of 1874 from Germany, from the cereal crops, and in India it is recorded from Rajasthan by Dr. Vasudeva in the year of 1958, and it spreads in Europe, Australia, Russia, Israel, India and Pakistan. In India it widely spread in Rajasthan and Haryana. Other than these two states, Delhi, Himachal Pradesh, Jammu Kashmir, Madhya Pradesh, Punjab and Uttar Pradesh also these nematodes are available. It will infest mainly on wheat and barley. In Europe it will infest on oat also. So in Europe it will infest on oat is recently identified in the oat. After 2000 they identified, and it caused molia disease of wheat and barley. There is two pathotypes of these nematodes are available in India. One is HA31, another one HA41, and it causes crop loss. In Rajasthan alone, this Heterodira avinae it will cause up to the yield loss of both wheat and barley in 8.2 to 28.8%. So how we can manage this nematode from wheat and barley means we have cultivate the non-host crops like mustard, chickpea, coriander, cumin. For 1 to 2 years we have to cultivate with the as a non-host crop, and summer plowing we have to do it for 2 to 3 times at the time of May, June, and yearly sowing. So here if we have any yearly sowing varieties of wheat and barley we can sow it yearlier. And resistant varieties for barley, Rajkiran C164 BH393, and for wheat Raj MR1 it is available in India, and Katli it is available in Australia, but this Katli variety is not it is a susceptible variety in India, but this variety is resistant in for Heterodira avinae in Australia. Then chemical method of control is application of carbofuran at 2 kg active ingredient per hectare. Heterodira gajani, this nematode it spreads in India, Pakistan and Egypt. It infests on the Kharif pulses. So Kharif season la where whatever the pulses will be grown in the season of Kharif, this nematode infests on those crops only, and it completes its life cycle into 3 to 4 weeks. It causes yield loss. Also, it affects the nitrogen fixation of the plants. In cowpea, this nematode will cause up to 20 to 28.6% of yield loss, green gram 38%, pigeon bee 23.3% of 7% of yield loss. So the management practices, we can cultivate the non-host crops like wheat, maize and rice. We can cultivate pigeon pea with sorghum as an intercrop, and seed dressing of carbofuran at 3% active ingredient. And potato cyst nematode, this nematode is identified by Kuhn from Germany. In India, it is identified from Dr. Jones in the year of 1961 from Nilgiris. Now it is also available in Himachal Pradesh, Kerala, Kodaikanal, Karnataka and Palani Hills also. It has 5 pathotypes. In both Globodira rostochiensis and Globodira pallida, there are 5 pathotypes are available. In India, Globodira rostochiensis RO1, RO2 and Globodira pallida PA1, 2, 3. These are the pathotypes available in India. It causes the yield loss up to 53 to 66% of potato. So the management practices, we can cultivate the non-host crops like cabbage, cauliflower, radish. The resistant varieties, we can cultivate the Solanum verni. It is multiple gene resistant, and another one is single gene resistant variety is Solanum antigena. And in India, Kufri Swarna, it is a resistant variety. And we can apply carbofuran 2 kg active ingredient per hectare. And in India, we have a destructive insect pest act for Globodira for prevent the spread of this nematode from other states. So other than these chemical methods, there is no much chemical methods are available for the management of nematodes. So we can go for the biocontrol agents. In those categories, Pseudomonas fluorescens, Bacillomyces liliaceus, Poconia chlamydophora, VAM, Trichoderma viridie. These are the biocontrol agents we can use for the management of nematodes, especially Bacillomyces liliaceus and Poconia chlamydophora. These are the widespread yet parasitic fungi, and it is widely used for the management of cyst nematodes in many states of India and also in foreign countries. So, thank you.